Well, thank you all for joining this CDDF webinar series, and a very topical subject tonight, how to analyse clinical trial data collected during the pandemic, with Dr. Jan Bogaerts from the EORTC. Can I have the next slide, please? So, as many of you online here know, CDDF is the Cancer Drug Development Forum. We're a non-profit organisation registered in Belgium, providing a neutral platform to stimulate interaction between all of us stakeholders involved in cancer drug development. So bringing together people from academia, the pharmaceutical industry, the regulatory authorities, policymakers, and very important patient advocacy groups. And you can look at the detail of what we do and what we offer on the website that's there. Next slide, please. Technical aspects, and I think as many of you know, the technical side is not my forte here. What we're suggesting is that there's over 100 of us online today, so if everybody's on mute, we get better audio quality. If there are any technical problems, please put them in the chat function and put questions in the Q&A, and then at the end of Jan's talk, I will facilitate a discussion of those questions as well. If you double click on the Zoom screen, you can go into a full screen mode. So next slide, please. So the agenda today is my brief introduction, and then more importantly, Jan's presentation, which is about 25 minutes, and then I'll moderate the question and answer session. But next slide, please. And really to introduce Jan, who will be known to many of you. I've known him for many years. He and I were just chatting about anniversaries for EORTC that are coming up. He hasn't been there all of the time because the EORTC is nearly 60 years old. He got his degree in mathematics in 1986 and then a PhD in mathematics in 93 from the Free University of Brussels, but joined the EORTC in 2004 and is currently its scientific director, and also serves as a statistician on the EMA Scientific Advisory Group for Oncology. And his interests are methodological interests, issues around progression-free survival, where I've worked with him, alternative ways of looking at change in tumour measurements as predictive markers, and how to correctly evaluate new markers in existing risk prognostication. But for the next slide, before we actually go into Jan's talk, we were going to do a poll, partly because I know Jan's very interested in the answer to this. So we're going to go into the poll function, and you'll see the questions there. You can only choose one answer, and I'm not allowed to vote. When you're being presented with trial data from a trial that has been influenced by the COVID pandemic, which would you prefer? To see the data as it happens, with some reporting of the COVID impact, to see an estimation of the effects as they would have been in the absence of COVID, or something between these two options. And we'd be really interested if you wouldn't mind voting on that now. And then we were going to repeat it after you've heard Jan's talk and seeing how, after he's spoken about some of these issues, whether that will influence what you feel. So if people wouldn't mind voting now, and I'll give you 30 seconds to a minute to do that. So I'm hoping, I can't see if we're getting the votes in, but I'm hoping we are. Yes, thank you very much. So should we, I think we've got, adding them up very quickly, a significant number of votes in. So let's close the poll there. And I think then, will that come up on the screen everybody can see? Yes, I think it has done. So we're a little bit split. Less enthusiasm for an estimation of what they would have been if. So either seeing the real data or something between the two, but with the estimation as well. So with that, I will close the poll. We are going to repeat that afterwards, but actually, without any more ado, was going to suggest we go into Jan's talk. So thank you very much.
Good afternoon. My name is Jan Bogaerts. I am a statistician. I have a role as a scientific director at EORTC headquarters in Brussels, and I will be talking to you about ideas and concepts around the analysis of clinical trial data that were affected by elements of the COVID pandemic. So in essence, what are analysis ideas you should be aware of and could use in reporting data that have elements of the pandemic? So first off, I want to show you this concept from my imagination, if you like, and that is an axis running from real-world-like analyses to clinical trial-like analyses. So in a clinical trial-like analysis, all the attention, I would say, goes to causality, usually for the randomized question. And there are very strong boundaries on the data that is being collected, and the data is strongly curated, highly compliant, and potentially, in my opinion as a statistician, all of this curation and fine-tuning and attention to detail can lead to a certain overestimation of how the treatment effect would be in a real-world setting, so later on in the clinic. That was the right-hand side of this slide. The left-hand side is a real-world-like analysis, which is using the data, potentially the same data, but from a descriptive, informative point of view, and that more accepts the situation as is, and there is less a priori selection. This, of course, can then focus on many outcomes, and it could, for example, be the secondary use of clinical trial data for other analysis purposes. In this vaguer approach, this could lead to potential underestimation of effects, the case occurring. So recently, all analyses in my mind are somewhere on that spectrum. So within that concept that I've just shown you, you can think for yourself where bias could originate. We're thinking about bias related to internal and external validity, but also biases because of unknown covariates that could confound any analysis. You could also ask yourself, what is the external view on these data that you have in mind? Because that could be interpreted in several ways. So at this point, I want to introduce the concept of estimates. Estimates are a bit of a new neologism, I think. I did not know it as a word before. There is an ICH E9 addendum on estimates and sensitivity analyses in clinical trials to the guideline on statistical principles for clinical trials. And the question then is, what is an estimate? Well, any statistical analysis will try to estimate quantities, in this example, a treatment effect, describing a difference in outcome between taking and not taking a treatment, or for example, two different treatments. And the whole terminology of estimates is trying to fine-tune that question to the point where it becomes clear to everybody why certain choices in analysis were made. So we need to agree what the exact treatment effect we are trying to estimate is. And before designing and analyzing the clinical trial, it should be clearly defined which treatment effects are of interest. The case we have here, where data has been affected by COVID, you're not at the design stage, I imagine, but you're in a situation where the data is as you have it, or has become as you have it, and you're wondering for yourself how you should analyze these data. I feel there is value in this discussion to looking at the problem from a point of view of estimate terminology. Why? Because it is not a revolutionary terminology, but it can give a lot of insight. Industry, health technology, and regulators are moving into this framework to clarify their exchanges of discussion with pharma industry and with others providing them with information. And I think it leads to transparency on decisions made with regards to statistical analyses. So yes, it is rather formal, but regardless of the formality of it, I believe it could help us understand how we would like to work in COVID-affected data, so to speak. So a bit more formality. What makes an estimate? There are 5 attributes, and you can read the guidance for yourself. There is the targeted population. Which population are we trying to estimate something for? There is the variable or endpoint to be obtained for each patient, which is the endpoint. There are the treatments. What is the arm? What are the arms? The specification and handling of intercurrent events, and that's going to be the crux of our discussion today. And then there is a population level summary that will be constructed out of all of these. These 5 attributes interact with each other, obviously, and we will see examples. So intercurrent events are, I would say, the main focus of this discussion, also as it goes into the COVID as an intercurrent event. And we could discuss later on which intercurrent events exist as related to COVID, because you could argue vaccination could also be an intercurrent event. Getting COVID disease could be an intercurrent event. Missing visits because of the hospital needing to redo its planning could be an intercurrent event. It's not a single intercurrent event in this situation. But in the general sense of estimates, things happen in a clinical trial, and the statistician and co-workers need to decide how they are going to deal with these. And we already have many examples of how these decisions are taken in the present and in the past. So you want to specify intercurrent events up front. You want to think about whether intercurrent events distort the interpretation or prevent the estimation of whatever it is you're trying to estimate. And depending on how you handle it, it could be that uncollected observations are not really missing, but you have another way to deal with them. For example, let's say the endpoint is response at the end of treatment and a patient stops early due to toxicity. What do you now do? You could say, I have a failure by definition. And then the statistician will say, well, anything that is missing afterwards is not a problem because I already have concluded a failure according to the rules. Or it could be that the rules say a further response assessment is required. In that case, you start having a missingness problem at the moment a patient stops treatment because of toxicity. So this is a point in time, and I will repeat this probably, where I want to ask you and to make you aware the possibilities of analysis in the presence of COVID-affected data very much depends on what you have in your data set. It could be that you only know that COVID was a thing during your time of observations. It could be that some sites or some hospitals have told you, well, we have a problem here. Or it could be that you have detailed data points on every patient, if they were vaccinated, whether they were vaccinated, if they got COVID, what happened to them, what treatment they were given. You could have all levels of data available relating to your intercurrent event, and your options of analysis obviously will be affected by the presence or absence of such data. So we now go into a funny slide, which I'm indebted to Corneel Koens, one of my colleagues. So the 5 proposed strategy in the new ICH guidance can be condensed as follows. The first one is treatment policy, and that is ignore as much as possible. That is the typical way forward that statisticians choose in comparative analyses of phase 3 trials, where they say we will do intent to treat. Everybody will be analyzed in the arm they were randomized to, regardless of what happened. So that is part of treatment policy strategy in analysis. A second option is composite. You make the intercurrent event a part of the endpoint, as we've just discussed. If a patient goes off treatment for toxicity, that patient is now a non-responder. That is an example of a composite interpretation of response. You could say in a progression-free survival analysis, if the patient goes off treatment, we will, without having progressed earlier, we will consider that a progression. That's an example of a composite policy. And the Smurf on the right there is exhibiting that. He says it looks like part of the event to me. The third option is hypothetical. I call that the counterfactual question. Please, dear statistician, you are so good. Please give me the results of your analysis if the intercurrent event was not a thing in reality. That's a very complex question, and you can imagine to do that, you need advanced statistical technology, and I will make some comments about that. The fourth policy is principal stratum. It is to try to limit to those patients who did not have the intercurrent event. That can be a reasonable strategy if you did not have too many patients with the intercurrent event, but it is at high likelihood of introducing bias into your analysis, because you will probably have the experience that your local statistician is not very eager to exclude patients from analysis because of that fear. And the fifth and final strategy is while on treatment, that is limiting to the period without the intercurrent event. So you know that type of analysis in time to event analysis, where the statistician decides to censor the patient at the time when something happens that prevented further observation of the event of interest. Let's move on, we will come to examples. So in the COVID situation, I will repeat my first slide. You can go for an interpretation that is, in my mind, more real world-like, that is more ignore, just describe everything you have seen. And that is, in this case, the fairly straightforward way. And you can go clinical trial-like, which is more hypothetical, the third option we saw, the counterfactual question, where you ask the statistician, try to tell me what would have happened without COVID. I'm not sure that's what you want to do, but we have a quiz question on this. So in this case, the clinical trial-like analysis is more complex, and then you have a higher need for sensitivity analysis. I want to highlight that that is my position about the complexity of analysis in this COVID situation and how you would analyze the data. In general, I believe that if you have clinical trial data, the analysis and the interpretation is more straightforward and more simple on the right side than to do a proper analysis of real-world-like data. So this is an illustration out of the EMA guidance, which is asking you to do sensitivity analysis. And I think sensitivity analysis, especially when you go into complex analyses, can help the reader or anybody who sees your results to better make up their mind how far the bias might be going. So I encourage you to do sensitivity analysis along with complex analyses. And the most obvious sensitivity analyses are showing the data as they actually were, so the Bruto data, and then show positive, optimistic and pessimistic scenarios for the data. So think of the following before you start planning your analysis. You will need, in my opinion, to evocate how far COVID has affected your data. Which hospitals were involved? Probably all of them, of course, you know that. How countries were affected, how calendar timing was affected. There were strong crises during the COVID period, and there were more extended periods when the situation was different. What was the capability of the trial people in the hospitals to cope or comply with your protocol treatment and whatever your protocol was asking? That could be very different depending on the trial, and we've seen that at EORTC. And you may try to summarize the deviations you have observed to the extent you have those data available. And you want to make a clear assessment of the extent of data that you have available related to COVID impact. So for example, we have concomitant medication recorded as it relates to COVID. You could state that, it would be very interesting background information. Or we know which of our patients contracted COVID disease. Very interesting information. In general, you want to assess to what extent all of this meant a deviation from your normal protocol development. And then for each endpoint, you want to identify the intercurrent events you want to take into account. as a general example, we could have tumor response as noted best RECIST response, let's say, at the end of treatment. And examples of intercurrent events could be death or treatment switching. And I will use treatment switching due to toxicity as an example in the following slide, how you could process that in an analysis. So here are examples with the policies that we've seen before of what you would do. In treatment policy, you would just ask to continue observation of response, and if response occurs, you will say this patient responded. You can go composite, you can say, I have a tumor progression at the moment I switch treatment. And then you include the fact of treatment switching into your endpoint. I'm not saying that's a good idea, but it is an example of the principle. You could go counterfactual, hypothetical, and you could do extensive modeling for any patient that goes off treatment because of treatment toxicity to calculate what would be the probability based on other patients in your trial that they still would have experienced a response. People usually don't do that, but it is statistically possible, but more risky because biases might creep in. You could go principal stratum, where you say, well, I'm going to limit my analysis to patients who were not affected by my intercurrent event. Your statistician will not like that, but we've seen plenty of people do that. You could do while on treatment, where stopping treatment is seen as a competing risk. In this case, it does not look advisable, but it can be done. For other endpoints, that is something we do. So I go back to the question, what makes an estimate? And as I said, intercurrent events, or I would say the core of attention in this methodology that I'm presenting here. And this is just a slide to tell you that the other composites of the estimate are affected by all the choices that you make. But I think that's kind of obvious. So maybe another superfluous slide. Why is COVID-19 an issue? Well, I'm sure that in previous talks about the topic, we have all been thinking about how it affected the data we are getting. It will affect, it may affect the outcome. It comes across the observations. So treatment may have been delayed, treatment may have had to be stopped. Assessments may have been missed, lab assessments, other assessments. Toxicity may have occurred, directly or indirectly related to COVID infection. And you want to ask yourself, obviously, how far am I bothered by this in making your plan to do the analysis according to a certain construct, as I've shown before? So here are some ideas you might think of doing, and possibly also some sensitivity analysis you might extract from this table. Here are some suggestions that we've thrown around internally at EORTC about how you could analyze data with COVID present. If you have a long clinical trial, you could have a part of the clinical trial that was pre-COVID, or might we say post-COVID at a certain point. So you could use general calendar time windows where you stratify your analysis, or you just cut your data in pieces, in chunks, in a pre-COVID period and a during COVID period. And then you can have a sensitivity analysis saying, well, I analyzed the data that was only pre-COVID, and then I analyzed the data during COVID, and I compare whether it looks more or less the same or rather different, and I will make some conclusions out of that. You could go further on the second line. You could say, well, I want to be a bit more subtle. I look by country, when COVID started, what were the measures, how deep the measures were, and so you could do a more refined option too. I think it's going to take you down some rabbit holes, but it could be done. I could even imagine that you have signals coming out of your hospitals. We are very much affected, we are okay, and that you analyze according to that. Of course, your later reader needs to be convinced by how you did it. And in that sense, simple is probably easier. You could go event-based. You may evaluate how much a patient being affected by at least one COVID-related event, whether to keep that patient or throw the patient out for your sensitivity analysis, either event-based or relevance-based. There you really need your medical, for the last category, you need your medical review to say upfront, before you start doing your analysis, your medical reviewer has to say, well, in this case, COVID played a role in how it turned out for the patient. That can, again, give you a very useful sensitivity analysis to see to which extent COVID affected your results. I will give you some statisticians always have concerns and headaches, so it's no miracle that I will give you some of the ones I'm thinking of. COVID disease status and how bad the disease is for an individual is clearly related to age, we all know that, and also to other factors such as smoking and obesity and gender, I believe. So statisticians will consider, will worry about confining with site and with country, and you want to look at that. You want to ask your analyst to take a look at the possible confounding. We will say in the second bullet again that if you start excluding patients, efficacy outcomes from patients without COVID should not be considered representative of the trial population, but it could be a very useful sensitivity analysis. AEs may be very difficult to distinguish between COVID or general disease-related or treatment-related adverse events. So it's probably going to be very hard for some adverse events to really dissociate. And you may ask yourself, is vaccination another intercurrent event that you want to give its own taste of further analysis? And maybe does it bring, will you do a sensitivity analysis where you consider the case or the patient to be brought back to a pre-COVID status? That is quite possible. Other issues I want to point out, there is the impact, things we did not talk about now, but you may want to take a look at as well in your reporting, the impact of recruitment, suspension, and reduction. How much was your trial affected in terms of needing to run the trial longer than planned? Has that somehow affected the general interpretation of your trial? That could have happened. In general, I don't believe it will happen, but it could in some circumstances. For example, in a time-to-event analysis, the preponderance of data coming to the time-to-event analysis, which is event-driven, could completely come from the earlier patients. So you could have a situation where your total number of events needed to do your comparison, your log-rank test, was created by the earliest patients because the trial so much slowed down to the extent that the later enrolled patients have not really contributed to the trial. That's something that really could happen. You could look into unblinding due to COVID-19. That could be a consideration. And, well, impact on health-related quality of life. That is another can of difficulties, I would say. So this is my last slide before thanking people. In summary, I would encourage you to carefully consider what it is you would like to extract out of your data set. And I offer to you the estimate framework to take a position, to say, I want to do a puristic analysis as if COVID had not happened on the right-hand side of my first slide, or I will prefer to generally describe essentially what has happened in the presence of COVID and then do some sensitivity analysis to try to see how far that might be from a non-COVID situation. You should consider what kind of information and details you have access to. I want to repeat that because that will define the limits of how far you can go in analysis or how far your statistician can go. And I encourage you to qualitatively and quantitatively report on the impact of COVID on your trial, on your data set. Then choose analysis strategies before doing the analysis, obviously, and they could be endpoint dependent. You could say for overall survival, I'm going to adjust my survival analysis for COVID deaths. I can see that happening. My response to treatment, I will adjust it for the availability of treatment. So you could have different strategies depending on the endpoint you want to summarize. And the more complicated the analysis, the higher is the need to embed the strategy of analysis in some sensitivity analyses. For example, the raw analysis, just what happened in the trial, disregarding the fact that COVID was ongoing, could be a useful sensitivity analysis to report if you do something more sensitive, something more complicated. So I want to end this talk by thanking the organizers for the very kind invite and for giving me an opportunity to think about this question. Our statisticians have all been thinking about it, and I don't claim to have all the solutions. I wanted to give you some options and some general guidance on how you could define your strategy of analysis and present it to other people who will look at your research. And of course, I want to thank my colleagues, Cornel Koens, who is, I think, responsible for the Stroofs in the presentation, Madeleine P. of our Quality of Life department, and the other statistical colleagues, Saskia Litière and Muriel Mauer. Thank you very much.
No, thank you very much, Jan. And that was really clear. And it is a huge issue. I mean, we all know that our trials have been affected, but how we're going to tease that out and starting to think about that as they mature, and we still want to be developing better cancer treatments for patients is really important. Before we move into the question and answer session, I was going to repeat that poll question, if you don't mind, and then we will get Jan up on the screen as well live. So here is the poll question again, if you wouldn't mind voting. To see if this is, Jan's presentation has actually changed your thoughts on what you might want to see. So if I give you about a minute to vote for this, again, those three questions: see the data as it happened, with some reporting of COVID impact, do the estimations of effects of the absence of COVID, or something in between the two. So I encourage you all to vote. We're coming up to the minute on my timer. So if we could close the poll now and get those results up. And from memory, I don't think that's changed much at all. I think it was 15% for the middle one before, and we were pretty evenly split as a group between the other ones. So if we close the poll to keep you all on track with where we are with this webinar, and so I'm hoping now those of you online can see both Jan and myself, and we move into the discussion session. But I please encourage, I can see there's 84 of you online, to put your questions in the question and answer bit, and then I can then field them and put them to Jan. But while people are thinking of their questions, Jan, I mean, while you were talking about intercurrent events and that things happen on clinical trials, I was thinking that's true. Bad things happen. We know that. I had a patient who was flying in for a trial who fell down the aircraft steps and broke her arm. So she ended up admitted with an SAE, completely non-trial related, and was thoroughly embarrassed by the event. But those sort of bad things are random, whereas this was global, I mean, truly global. And we all collaborate across Europe, and many of our trials sit across the States as well. So we had a bad thing that happened globally. And can you model that in, or is there too much difference between countries, and do you need that level of detail?
Good evening, everybody. I think you actually might have situations where you want to be rather ambitious in your estimation, and you have a strong desire to know what would have happened without COVID, and where the most honest answer from the statistician is, we cannot do that for you. I did not say that in the slides, but I believe it is possible that there are trials that will be inconclusive. Another danger area is non-inferiority trials or equivalence trials, where the signal may have been diminished by the COVID situation, and where there is a strong danger that non-inferiority or equivalence is concluded because the signal has weakened. So that is quite a possibility.
And given where you and I have been working together, the thing that occurs to me is that we were up in the north of England more protected than other areas, and actually kept our trials open. But where we struggled was getting scans. So we've slipped on our scan timing. And particularly in later phase trials where those scans that often are driving endpoints, both progression and thing. So that's a sort of unknowable to model in, particularly as you highlight, a non-inferiority trial, isn't it? If there's been a pushback on some of the scans.
Yes. You can make reasonable estimates from the data. That I think your statistician can always do for you, because we have the statistical technology to try and do that. But that's what I wanted to point out in the presentation, that you first have to make up your mind what it is you most would like to get out of the situation. I had the intent to not be prescriptive and tell people do this or do that. That was probably very clear from my presentation, but to give ideas of how you could handle it.
Yeah.
But I guess that I was not committal in what I was saying.
Yes.
I was very interested to see that the poll shifted a bit to the mixture response. So there were 50% of people on the first poll who said, just give me the data and I will look at the data. And a few more were answering, well, I would maybe like to see a mix. So there was a little shift in the answers, but nothing major.
I was going to say, there's a true statistician. I just looked at the numbers and said, yeah, they're roughly the same. He spotted the difference. And we've got a question coming on the chat from Diraja. I'm going to mess up all my words here. Diraja Gambaya, asking, at what point magnitude of impact of COVID such an analysis will make sense? How would we decide when do we do the magnitude of impact of COVID? I think I'm not sure I'm fully understanding the question, but I would think any trial that ran for a period of the time of the COVID presence will need some analysis along these lines, because people worry a lot about it, I think, and rightly so. We've seen trials that were seriously hampered by the situation. Yeah. So I think you need to do something. I don't think there is a particular threshold, but what might be very attractive is that you make an estimation together with your investigators to say, during what period do you think the trial was really handicapped or made difficult by the COVID situation. I somehow seem to have a belief that in terms of operating hospitals, and you can confirm that better than me, there's probably a time point where things went more or less back to normal in terms of doing the protocols that we want to do. So maybe it is possible, and that was suggested in the slides, to identify a core period of time when things were hard. And that's a good point to do sensitivity analysis, of course. I mean, the other challenge is that, particularly with trials where there's a randomization against two treatments, is one of the many unknowns we've got is the effect if patients in a particular arm got COVID, whether that had an impact simply because, I mean, we know at the beginning there was very significant worries about what would happen to patients who were on treatment who got COVID. In the solid tumor practice, and we actually saw less of an impact if we've been tracking and looking at outcomes of patients in the UK on more normal chemotherapy and immunotherapy, and targeted agents seem to have had the same pattern of COVID than patients not on treatment. But we didn't know that, and we don't really know it in a trial setting, do we? Whether or not there are now going to be differences in the. So presumably, what's going to be key as we start to try and analyze these trials that run over the period is to know who in which arm got COVID and have that data collected.
Yeah. Well, as I said in the presentation, you may not know. It depends on what level of detail you were able to collect, of course, how far you can answer such questions. The other thing I'm a bit worried about is that it is possible that COVID affected the treatment being given in one arm more than in the other arm.
Yeah.
That's a special concern for comparative trials, because in general, in randomized comparative trials, you could make the rough statement that if the treatments themselves were not strongly related to how we reacted to COVID, the randomization will mostly take care of things so that on both arms, things happened, but the comparison is not too much disturbed by that. That would be a bit naive way of saying probably it's okay. But you just heard me worried about equivalence trials that might falsely look equivalent because there was too much other things going on, making everything look the same. But once you start, I worry more about that than about a superiority trial not looking superior. But of course, clinicians might see that very differently.
But one of the other issues is that the randomization, if you've got multiple centers, won't necessarily, when you randomize, take into account the center. So you could end up with a skew that places that were more badly affected by COVID happen to recruit in a particular arm. And then you've built in that inherent bias, haven't you? When it becomes very complicated to tease out the COVID effect on those data.
Well, if you would exclude a number of centers that were most affected by COVID, if that would be your situation, and you would exclude them in a sensitivity analysis, that randomization would still be valid in the other centers, even if it wasn't stratified for center. That's perfectly possible. And I think for anyone looking at the results of the analyses, it's going to be very telling to see, okay, did they really see a difference when they did those sensitivity analyses? I think I really want to encourage people to do a few sensitivity analyses to see how it might have been different.
Yeah, and we've got another question in the chat from Li Zhao saying, if a subject missed a few scans due to COVID and later is found to have progressive disease, how would you handle that one in a progression-free survival analysis? And do you consider missing scans as an interrupt event? And can you then hypothetically model a hypothetical strategy be applied to that? This is a lot of complex stats, I think.
Yeah, you can do several things. I have to be a bit technical now. You could do the naive way, the carry forward, where you take the progression event at the time you see it, knowing that it could have happened quite a long time before.
Yeah.
You could, if you have a lot of that, you could do interval analysis, interval survival analysis. And interval survival analysis only uses the information that the progression happened between time point A and time point B, and that might be a long time. So that's a special kind of analysis that is called interval censoring. Or you might apply the famous table A from FDA that says if you miss 2 visits, you censor the patient before they miss those visits. But then, of course, you might lose a lot of information. Those are 3 options. None of those really takes care of the fact that the patient might have missed treatment as well. Because it's one thing to miss the scans, it's another thing if they have missed treatment and they're in that period of time progressed. Then it becomes very complex, of course, because it becomes a chicken and egg thing.
Yeah, no, it is. I mean, I guess a lot of this is going to start coming out as we clean the databases, close out the studies that have run over it, and then see, because I think we still don't really know what the impact has been. We know there's been a huge impact. We know there have been, as you say, periods where things really went quiet, but then people tried to get restarted. And we're all waiting to see what the winter brings. So I think we're not quite out of the woods yet with this. But how do we then, because we still need to develop better cancer drugs for our patients, and therefore we still need to try and keep these trials going. And you also have a responsibility, in my view, to the patients who've gone on the studies already to try and do our very best to, what they've put into it. make sure we get the data in to analyze.
Indeed. And in terms of coming with a convincing analysis, I would say now is the time to make up a plan, to make amendments to your statistical analysis plan, and then do those sensitivity analyses when the time comes, rather than first look at all the data and then say, oh, we're going to do it this way or we're going to do it that way. Any statistician will tell you that the first option, to plan now and do the analysis when we've locked the database, is the more convincing one, obviously. Something I have not touched on, if you are really worried that COVID has hampered the comparison or the conclusion of your trial in a select number of patients, the most drastic solution is to over-enroll when you feel COVID is no longer an issue, to make up for the numbers that you've lost during the COVID period. I don't think that's going to be a very popular one, but that's the most drastic option.
But when we think about the comment of seeing some of the some of the data that was in your poll question, you mentioned partway through that if you do a time window analysis, specific analysis, that you use the phrase that you could go down a number of rabbit holes. Are you really saying that that's just going to be so complex because this has affected different places in different ways, and how would you choose your time window? And yes, as you say, we had a period last summer where things were better and then it got worse again, more in some countries than others, and some places are having a third wave. Is that what you were meaning, that this is possible, but that's a lot of statistical time and maybe that you don't answer a question?
It's going to be very complex, I think, but it is still possible to exclude certain chunks of time. But of course, how to decide, because there's not going to be someone from WHO going to come and say, do it like that. That never happens. I think people just have to do the best they can with the data they have. But we've all seen those curves of the waves. So maybe there is something there that we can use. During those periods, our hospitals were seriously affected, so we can adjust the analysis to that.
And I think one of the other challenges, even around Europe, but it also particularly including the US, is the very different way different countries have reported COVID numbers and COVID deaths. And it seems to me that the only way we'll try and tease this out is when we are finally coming out of this and being able to think about other things properly, is looking at deaths over expected deaths. But some of those will be due to the fact that patients with cardiac problems didn't go to the hospital on time. So, there will still be non-COVID deaths in that, but I think it might give us a better handle on how many there have actually been, which I think we really are still struggling with the numbers.
Yeah, that comment rings very strongly with me, being a Belgian, because we were the record holder for a time and our newspapers kept saying it's because we are so puristic. So yes, that was a strong emotion for us. Yes, in a general sense, epidemiologically, the final answer is in the excess deaths that you see, and that is the true answer to, well, what did COVID do to the situation? Of course, there's also other COVID effects like long COVID that is reported to be a real problem. And I know people are trying to do studies about long COVID. It's not only the deaths that make a difference, I would say. There are other effects as well. My talk, of course, was about trying to do a good job of interpreting the clinical trials that we've been running in the period.
Yeah.
And there is no ideal good way. But we have the technologies that we've always used and that I've tried to explain. We've always been doing this in certain areas where things always happen in trials and we have a way to deal with them. But indeed here it was something very broad and very specific. So I do believe sensitivity analysis will help to ascertain to what extent it really made a difference for the interpretation of the trial. And as I said at the start of the live exchange, there may be studies that will remain difficult to conclude from. There may be such studies.
Yeah. No, well, I think we're coming up towards the end of time, so thank you very much. For takeaway, and probably you should patent, were those wonderful Smurfs. I like the Smurf that just says, that's life, I don't care. I'm going to do an intent to treat analysis. I think that sums up intent to treat analysis for me. But that was a lovely imagery on what those various methods that we could think about using. So I think you probably need to make that into a poster to share. The EORTC...
Credit goes to my colleague Cornel, because he is a well-renowned Smurf specialist, and so I don't want to take his light in that. I felt that the slide was so nice, I wanted to put it in.
Yes, but I think we're both showing our age. But with that, thank you again, Jan, for doing us and also being such a great sport over getting this organised as well. And I really do hope we get to meet in person next year when we're next scheduled to meet. So thank you very much indeed.
Thank you very much, everybody, including the organisers, of course. And if people still have questions, feel free to mail me or to mail to the organisers, and I can still give a shot at answering those. Thank you very much.
Thank you. And then to wrap up, to thank everybody who's dialed in tonight and participated, and to also to flag up some upcoming meetings we've got with CDDF. I'm chairing the one which is now next week, frighteningly, on digital tools and artificial intelligence in the oncology drug development. I think we've got a great lineup of speakers, and I'm very excited with some of the pre-recordings and talks that I've already seen. So that's the online workshop. And then in November, and we're still hoping this might be a hybrid event with some face-to-face element. This is Gene and Cell Therapies in Oncology, a workshop that's been organised and we're hoping will be in Amsterdam. And headline here, the CDDF Annual Conference for 2022. And the plan is a hybrid meeting, but with as many as possible as we can manage of us on site in Noord-Zee in the Netherlands, without a sandstorm this time, unlike 2020. So with that, thank you all for joining the meeting. Thank you to Min and the organising team who kept me right. And I look forward to seeing you at the next CDDF webinar. Thank you.